Welcome to Smart Biology Teaching. We are going to see about a topic that is protein modeling. This is an important topic in bioinformatics. We'll see about the description and definition of protein modeling. It is simply known as computational protein structure prediction. The definition here is the prediction of three-dimensional structures of protein using in silico techniques. The basic principle of the modeling relies on the known structures which is obtained using X-ray crystallography and NMR spectroscopy, where previously X-ray and NMR were the only techniques to be used in order to resolve the structures of the protein. But nowadays, advancements in the technology has brought about so many other techniques such as solution NMR, electron microscopy, neutron diffraction, solution scattering, solid state NMR, electron crystallography, fiber diffraction, EPR, theoretical model, powder diffraction, and fluorescent transfer. From these techniques, you can resolve your structure, and these structures can be used for your modeling procedures. So there are three main methods of modeling. First method and the foremost method is the homology modeling. Second method is your threading and the fold recognition. Third method is the ab initio method. And in first method you also call homology modeling as comparative modeling, where you predict your structure based on the homology of your target sequence with your template structure. We will see about in detail about homology modeling in the next coming slides. Then the second method is the threading or the fold recognition method, where you can predict the structures of the target protein using known protein folds which is available from different databases. So you have different databases which is freely available, and you can access it, and you can download the protein folds, and you can predict your homology model. Then you have your third method, that is your ab initio method. In ab initio method, this method will be followed only if you don't have, in absence of your similar structure, this method is followed, and it is used to, and the protein structures are built based on the calculations of the favorable energy conformations. Among the three methods, homology model is preferred to be the most attractive because nowadays you have so many advancements in the technologies. So these technologies will provide you the crystal structures, and it is posted in structure databases. You can download it and you can use it for identifying a homology model of a unknown sequence. So we will see about in detail about homology modeling in this session. So you see that homology model is an prediction of structures based on the homology of your target sequence and the template structure. So in this case, you have your target sequence or the unknown sequence for which a model needs to be predicted. So in this case, what you do is the model structure should be similar to the model sequence should be very similar to your structure of the protein, and it should be more than 30%, showing 30% of identity. So when you map your target sequence with your template structure, it should be over showing over 30% of identity. For this, the tools and the databases are used are BLAST2 and the PDB protein data bank. This is the structure database will provide you the structures. The main tools or the softwares which is used for homology modeling is Modeler. This is supported with Linux, and it is also supported with Windows. You have a Python open script which you can download it, and you can also use it as a source code. Currently, GUI versions of program is also available in the name of EC Modeler. So this is the schematic representation for performing modeling. So first step is you have your target sequence. This is your unknown sequence. Say for example, this is your target sequence. Then you have your template to be recognized from the protein structure database, that is PDB, where this is your target sequence. This is your target sequence, and this is your templates being recognized. Then what you do is you align your target sequence and your template structure. So the sequences will be aligned and the backbone will be generated. After generating the backbones, you have the loop modeling method where you design your loop. Then you have your sidechain modeling for accurate or the exact structures. Then you go for your model optimization and validation using online web servers, which is already available. If the software is being supported with the model optimization and validation, then you can go with your software itself, or else you have your own online servers which will do the task for you. So we will see in detail about the steps how to perform modeling. So you have your target sequence. So target sequence should be selected, or the choice of the target sequence is the is the sequence length which you are going to preferably take into account, which will reduce the background interference when you are designing your homology model. So you generally call this sequence of your choice is the target sequence, and then you can either you need not, when you say for example you have your 1000 amino acid sequence and you want to study about only the domain of that particular, there is a SN which is going to play an essential role in that particular protein. In that case you can choose only the domain, and then you can go for modeling. In a 1000 amino acids, 200 amino acids going to be a domain structure, or 100 amino acids is going to be a domain structure. Then you can go for modeling of that particular domain, and you can study your particular functionality of that particular protein, or your research interest can be applied. Then you have your target protein recognition, and you are supposed to, when you are downloading your template structure from the structure database based on the template recognition, you are supposed to prepare your protein also. So this reference protein which you are going to take is called as the template protein, and your choice of what you are going to do going to perform the homology model is going to be your target sequence, and this reference structure is known as the template protein. So these two terminologies will be frequently followed, so it will be helpful for you to understand what is a target and a template in this particular slides when you are using. So this template structure will be retrieved from the protein structure databases. It could be any database, but most preferably will be going for PDB database. Then based on the highest identity of your target sequence and the template structure, we have already seen it should be more than 30% of identity. When this type of 30% similarity is not found, then you can go for other methods like ab initio, or you can go for threading methods. But where mostly nowadays we get the proteins. Previous cases we had the difficulty in getting the protein structures, so we adapted so other methods also. Then the choice of the template for protein preparation will be based on the relevant constitution. Say for example you have multi-chains, one protein will have multi-chains like A and B and C and D. So your choice of the chain, then water molecules, ligands attached to it. When they are doing crystallization, they add water molecules and ligands and so on. So all these complexities should be taken care, and you are supposed to prepare your protein, and then you are supposed to take it to the next level. So next step is going to be your sequence alignment, where you are going to align your target sequence and the template protein sequence, and using sequence based alignment algorithm. So you know about the BLAST tool, where BLAST is a basic local alignment search tool which will have different other programs. Like you have only your nucleotide sequence and you want your protein sequence. In that case what you can do is you can give a search as BLASTX, and where you will get your protein sequence. And likewise you can preferably use such programming tool for which you can get your desired structure. So the structure is going to be from your structure database. So the option should be selected as your structure database. Selecting an appropriate alignment algorithm is requisite for accurate model of your protein. So you are supposed to select the accurate programming tool. Then you are supposed to go for your alignment, and identical vicinity is the of that particular protein. So where are all the identical regions in that particular protein between the target and the template will give you the accurate model. Then prediction of the secondary structure. So say for example in a protein sequence you have your beta hairpin, and when you are modeled and you don't have your beta hairpin, so you generally have to see about the predicted secondary structures needs to be compared, and you have to see them. So this will help you in getting the accurate protein structures, and the functionality, mostly the functionality may not change. So in that case you are supposed to see about your secondary structure. So when you are modeling, you are using the structure prediction tools for secondary structure prediction tool, which is freely available from Nextbasic portal. Then you can compare your secondary structures of the target and the template protein and then analyze them. Then you are finally building your model, and after building you are going to make your loop modeling. So you have your backbone generator which can be visualized using a 3D structure tool, but you don't have the you are supposed to optimize your loops, and loops are always going to be bringing you the variation where you have the identical vicinity region. This vicinity region will generate the backbone, but whereas you have some variations in your template and the target, the sequence identity percentage will be missing in those regions, will generate a loop. This loop will be may have some variations which can bring about interference when you are going for your next level of drug discovery study, say for example. In that case it can bring you some difficulty. So you are supposed to optimize your loops. In that case you can go for the software that is the mod loop software, and you have other softwares also to perform. You can you can have those softwares in order to optimize your loop. Then you can improve the accuracy of the software structure using your loop modeling style. Then you are supposed to validate your model and optimize your model. So for optimizing you have to go for energy minimization steps, which is available within the software itself. So you can give your energy minimization methods, you can apply it, and then you can get your energy minimized structure. Then you can validate these softwares using you can validate these structures using the verification and the tools which is available, freely available. For this you will be using ProCheck, what if, all these types of programs will help you in validating your structures, and this validation test will provide you the energetically satisfactory model, and this model can be further used for your experimental approaches. So hereby I end this particular slide, and next slides we will be seeing about other topics related to protein modeling. Thank you.